Hello, my name is Thomas O'Connor, the Director of the Division of Product Quality Research in the Office of Testing and Research in the Office of Pharmaceutical Quality. In this presentation, I'll be discussing quality considerations for continuous manufacturing of APIs. This presentation is a collaboration between the Division of Lifecycle and New API in the Office of New Drug Products and the Office of Testing and Research. I want to start off by acknowledging all the co-authors listed here: Angeline Bain, Yoon Wang, Ram Ranadad, Dave Skanke, Adil Mohammed, Ray Frankowicz, and myself. Start off with some background. FDA recognizes that continuous manufacturing is a novel technology for the pharmaceutical industry that has the potential to increase the efficiency, flexibility, agility, and robustness of pharmaceutical manufacturing. Continuous manufacturing can improve pharmaceutical manufacturing by, for example, using an integrated process with fewer steps and shorter processing times, requiring smaller equipment footprint, supporting an enhanced development approach, i.e. quality by design, and use of product analytical technology and models, enabling real-time product quality monitoring, and providing flexible operation to allow scale up, scale down, and scale out to accommodate changes in demand. Also, for drug substances, continuous manufacturing can enable new synthetic routes and improves safety, health, and environmental performance. In 2019, FDA published the draft guidance, Quality Considerations for Continuous Manufacturing. For the purposes of this guidance, FDA considers continuous manufacturing to be a process in which input materials are continuously fed into and transformed within the process, and the process output materials are continuously removed from the system. Although this description can be applied to individual unit operations or a manufacturing process consisting of a series of unit operations, as described in this guidance, continuous manufacturing is an integrated process that consists of a series of at least 2 or more unit operations. The scope of the draft guidance is focused on solid oral drug products, based on FDA's experience with the review and approval of 6 regulatory applications utilizing CM for finished dosage form manufacturing. In addition, though, FDA has recently approved a regulatory application using CM for active pharmaceutical manufacturing and has had numerous interactions with sponsors developing CM processes for API manufacturing through CDER's Emerging Technology Program. Based on these recent experiences over the last couple of years, this presentation will describe how the FDA's current thinking on scientific principles described in the draft CM guidance are also applicable to continuous manufacturing technologies for APIs. This presentation will cover quality considerations related to CM for 5 different specific areas. 1. Characterization of process dynamics. 2. Process flow diagrams with sufficient detail. The definition of batch size. Process monitoring and control, including process analytical technology, PAT, and equipment and systems integration. Lastly, the presentation will conclude with description of how FDA's science and research initiatives support the development of quality considerations for emerging technologies such as continuous manufacturing. Product and process understanding form the foundation for effective risk management. The expectations regarding the science and risk-based approach to the control of processes and product quality based on process understanding are the same for continuous manufacturing as for traditional batch manufacturing. CM processes are dynamic processes that are subject to transient disturbances during normal operations. Larger changes in process parameters or quality attributes can happen when processes are in a transient state, such as during startup and shutdown, or change from one operating condition to another, or when a significant deviation occurs, such as those due to equipment failure or unexpected change in material attributes. The risk assessment for a continuous manufacturing process should consider the understanding of the process dynamics of the integrated system in addition to each unit operation. A suitable scientific approach should be used to characterize how a material flows through the process. One common approach is the characterization of the residence time distribution, the RTD, for individual unit operations and the integrated system. An RTD is a probability distribution that describes the amount of time a mass or fluid element remains in a process and can be measured through tracer experiments, online process measurements of appropriate product quality attributes, and/or modeling approaches. The shape of the RTD reflects the degree of axial dispersion or backmixing within that system, which affects the propagation of disturbances, material traceability, and impacts the development of the control strategy. The RTD should provide sufficient time for key transformations in the process, such as chemical reactions in the API process. The RTD is depending upon several factors, including input material attributes, mass flow rates, process parameters, equipment design, and operation. One key process parameter to focus on are the material flow rates, because flow rates will directly impact the process dynamics, and in a flow synthesis process also controls the stoichiometry of the reactants. It is important to understand how the RTD varies over the planned range of operating conditions, in addition to characterizing the RTD at nominal and target operating conditions. In API processes, different segments of the process can have different process dynamics, such as coupling of a fast reaction step with a slower reaction step downstream. Surge vessels can be used to decouple segments of the process train, allowing different segments to have different flow rates, and simplifying operational aspects, such as sample collection and maintenance. The surge vessels typically have a significant impact on process dynamics and need to be incorporated into the analysis. CM for API can involve hybrid processes that incorporate integrated CM operations as well as traditional batch operations. To support the quality review, sponsors should submit a process flow diagram with sufficient detail to illustrate continuous flow operation elements, such as the material flow, including hold-up steps, labeling CM batch processes, and identifying surge vessels, as well as identifying recycle loops. Identify the entry and exit of all materials. Critical process parameters and ranges, especially for flow rates. In-process controls, sampling for at-line or offline analysis, and PAT locations. If advanced process controls are used, such as feedback controls, those should also be indicated on the flow diagram. A continuous manufacturing process is expected to maintain a state of control and produce product within the desired quality throughout the manufacturing process. However, the manufacturing process will include periods when non-conforming material can be produced, such as during startup, shutdown, or temporary process disturbances. If approaches for material traceability, process monitoring, and material removal are well established, this non-conforming material can be segregated and removed without affecting the rest of the batch. Thus, in-process controls that lead to process rejection or diversion points should be identified, along with those diversion points. Reaction conditions and whether telescope reactions are present should also be identified. Telescope reactions are when materials are subject to successive chemical reactions without intermediate purifications and can be a feature of integrated CM processes. Shown here is a schematic of the continuous processing train equipment used for a recently approved multi-stage process. The process here consists of a series of integrated reactions and purification steps to synthesize the API, and then downstream batch crystallization steps are used to produce the final drug substance. The process has two diversion points located in red circles, so after stage 1 reactions and in the holding tank after stage 3. The process also has a surge vessel that helps couple the stage 1 reaction steps with stages 2 and 3. The picture shows how this multi-stage process fits into a single room. The definition of a batch has regulatory implications, particularly with respect to current good manufacturing processes, product recalls, and regulatory decisions. The CFR definition of a batch specifies a specific quantity of drug or other material that is intended to have uniform character and quality within specified limits, and is produced according to a single manufacturing order during the same cycle of manufacture. The CFR and ICH Q7 batch definitions are applicable to continuous manufacturing. A batch can be defined based on production period, quality material processed, quality material produced, or some sort of production variation, such as lots of incoming raw materials. If a time interval is used to establish a batch size, the corresponding input rates should also be provided to make the connection to the specific quantity of the API produced. When a batch size is defined as the quantity of output material, a suitable quantitative metric should be defined to establish batch-to-batch consistency and system robustness. Batch sizes can be flexible. Regulatory applications can include a range of batch sizes. For example, 10 to 40 hours. If a range is proposed, it should be justified, and the method of achieving the range should be described. Any proposal to change the mass flow rate to change the batch size should be supported by the appropriate data. The actual intended size of a given batch should be defined before the start of manufacture and be managed under the pharmaceutical quality system. The development of successful control strategy for CM is enabled by a holistic approach, considering aspects specific to CM and principles described in ICH Q8 through Q12. Process monitoring and control support the maintenance of a state of control during production and allow real-time evaluation of system performance. Common approaches for process monitoring and control including certain target set points and ranges, design space, control limits, and specifications for attributes being measured are applicable to continuous manufacturing. Critical process parameters that should be included as part of the process monitoring approach include flow rate, which impact both process dynamics and stoichiometry. Because of the impact of a disturbance is related to the magnitude and the duration of the disturbance, in-process control limits may include both a time and a limit and a time duration, such as plus or minus 10% from set point for 20 seconds. The material diversion and collection strategy should be described and justified as part of the application. The approach and criteria determine the time for and the amount of materials needed to be diverted and the basis to resume output material collection, i.e. the number of controls that were within requirements, should be described and justified. Factors such as the sampling frequency, the RTD, amplitude and duration, and propagation of disturbances should be considered in developing the diversion strategy. The amount of material diverted should be appropriately incorporated and justify safety margins, including the uncertainty of any other measurements. Procedures for managing material collection, diversion, and disposition do not need to be included in the application, but should be maintained within the pharmaceutical quality system. In continuous manufacturing, the time scale from going from starting materials to the API is reduced. Thus, traditional in-process sampling and analytical testing may not provide information on the quality of the in-process material in time to prevent any potential poor quality product from continuing through production. The utilization of PAT is well suited for CM to address these issues. In API processes, this includes non-specific approaches such as conductivity that monitor the consistency of a process step, such as a reaction, as well as more specific approaches such as online HPLC and UPLC that can provide detailed information on the impurity profile in real time. Instrument aspects for PET methods should be considered, such as interference due to flow, time of acquisition versus flow rate, the number and location of probes, probe failure and probe maintenance, etc. The development of process monitoring approach should include a risk assessment that includes consideration for how lapses in process monitoring data collection, i.e. recalibration of a near IR probe, may affect product quality. The process monitoring approach should be selected should include alternatives or additional quality controls to mitigate the risk to product quality posed by these scenarios. The use of PAT enables high detectability of disturbances in real time. Therefore, CM is readily amenable to process control strategies such as active controls, feed-forward, and feedback control, where process adjustment based on frequently measured or predicted outcomes enable the maintenance of a state of control. When implemented, supporting information and rationale for advanced process control approaches, including identification of the controlled and manipulated parameters, should be provided. Monitoring of a continuous manufacturing process using PAT tools can generate a large amount of real-time process and quality data during production, which can be used to support real-time release testing. Although real-time release testing is not a regulatory requirement for implementation of continuous manufacturing, it is encouraged and can be applied to some or all of the finished product quality attributes tested for the release of a batch. When real-time release testing is adopted as part of the control strategy, special considerations should be given to the sampling strategy. The selected sample size or frequency should be representative of the batch and should be justified using an appropriate statistical approach with respect to quality assurance. For data collected at high frequency, statistical methods for large sample sizes should be applied to provide improved characterization of the batch. Furthermore, procedures should be developed to establish a plan for real-time release to address potential gaps in PAT data, such as failure of a PAT instrument. For submission of NIR or spectroscopic PAT methods, refer to the guidance for development and submission of near-infrared analytical procedures. The scope of model development, validation, and maintenance, and details provided in regulatory application should be commensurate with the model type and impact. Refer to the points to consider for ICH Q8, Q9, Q10 implementation for regulatory expectations on models. Individual pieces of equipment, their connections to one another, and monitoring control systems and spatial layouts constitute an integrated CM system. The design of equipment and their integration to form a CM system impacts process dynamics, material transport, and transformation, output material quality. When developing a CM process and a control strategy, it is important to consider the factors of both the individual equipment and the integrated system, which can affect, for example, the ability of a CM system to maintain a continuous flow of input and output materials, manage potential disruptions to CM operations, for example, filter changes. Furthermore, appropriate design and selection of equipment for a CM process may enable process simplification or improve process capability and performance. For example, in drug substance processes, careful reactor design can effectively reduce formation of impurities, resulting in fewer purification steps. Manufacturers using continuous manufacturing processes may need to run equipment for longer periods of time to achieve the predetermined batch size. Equipment performance could decline gradually during the same run or after several repeated runs due to fouling or normal wear and tear. Such a performance decline may not be observed in short development runs. Therefore, equipment for continuous manufacturing warrants the following additional considerations for qualification, maintenance, and cleaning. Solids may be present in the reaction as reagents, intermediates, byproducts, or as the product. Lack of convective mixing and small reactor cross-sectional area in continuous flow systems may increase the probability that solids clog in the channels. Equipment qualification should address both the individual unit operations and the integrated system throughout the product life cycle. The development and maintenance of the control strategy should take into account equipment failure modes to ensure that abnormal equipment performance is detected and investigated, including appropriate corrective action. Equipment maintenance and calibration procedures should be developed and updated based on ongoing monitoring of the equipment performance and other available information. The process monitoring strategy should include indicators of equipment performance based on the qualification experience and understanding of the potential failure modes. This can also help address the maximum runtime for the integrated line before maintenance or cleaning is needed. Now I'd like to briefly touch upon how FDA's science and research programs support the development of the quality considerations for CM for drug substances that have been discussed throughout this presentation. FDA has developed several manufacturing platforms such as continuous flow and continuous crystallization to advance the regulatory science for CM for drug substances. Studies using these platforms have advanced the understanding of the risks to product quality, such as incrustation and solids clogging, as well as the development and validation of PET methods, such as Raman for concentration measurements and FBRM for particle size monitoring. In addition to internal research, CDER's capacity to address this complex area of research is further advanced by cooperative research grants and the Broad Agency Announcement Program, which allows FDA to tap into external knowledge and infrastructure in industry, academia, and other government agencies. As an example of this external collaboration under the BAA program, a CDER project has resulted in the development of a modular, integrated, end-to-end continuous process for a model API. End-to-end continuous manufacturing is a fully integrated process in which raw materials or chemical intermediates are continuously fed into the process and transformed. Then finished drug products are continuously removed from the system. In end-to-end continuous manufacturing, the drug substance is not isolated and therefore cannot be characterized by offline testing. The project demonstrated and validated several methods, including spectroscopic and process models, for monitoring and controlling product quality of the drug substance and drug product in real time. Knowledge gained from these internal and sponsored research programs inform policy, assessment, and inspection activities, ensuring that FDA's regulatory policies reflect the state-of-the-art manufacturing science. In conclusion, I just want to end with the notes that FDA recognizes and supports the adoption of continuous manufacturing as an emerging technology to modernize the pharmaceutical sector, to help patients by reducing drug quality issues, lowering manufacturing costs, and improving the availability of quality medicines. Quality considerations described in FDA's draft guidance on continuous manufacturing can support the implementation and regulatory submission of continuous manufacturing technologies for APIs. Additional references can be found here, which include guidances, as well as some papers referenced throughout the presentation. Thank you. Please send questions regarding this poster to the above address, and follow on webinar for both posters and presentations on April 9th. Questions can be sent there by email by March 19th for the webinar. Thank you.